Any questions before I move on there? Okay. So this is transfer RNA, or tRNA. tRNA molecules translate the mRNA code, linking the specific bases on mRNA. So the specific bases on mRNA there are on the bottom, and the tRNA is like a bedazzler. Like, it latches things on to the mRNA so that we can make these amino acids at the attachment site. So on the mRNA is a set of 3 base pairs, and the tRNA comes along and matches up to those base pairs and then makes the amino acid. So attachment sites consist of 3 base sequences that match up to the 3 base sequences on mRNA. Each 3 base sequence in mRNA, called a codon, whenever you hear the word codon, think of a triplet, those 3 parts right there, always matches with tRNA that carries one particular amino acid. So it's attaching, and it is holding that amino acid right there in the specific place so that it will be ready to build up into a piece of protein. So this looks crazy, but let's start in the upper left-hand corner. So recognize and initiate protein building. We've got our strand of mRNA that's right there, and it's got what's called a start sequence. So it's signified by the bases A, U, and G, and it's recognized so that the tRNA knows to come in at that specific place, at that specific starting point, and begin attaching the amino acids. And it is the attachment site of the tRNA molecule binds to the mRNA as the ribosomal subunits assemble around them. So you can see your mRNA, you've got your start sequence, which is the little green spot. You've got a large ribosomal unit and a small ribosomal unit, so they basically clamp down on the mRNA, and they begin working their way across, adding in those amino acids. And the tRNA are just floating around in the cytoplasm, waiting to get attached at that specific spot. So we've got like working down a rope. If you have a pair of gloves on and you're running your hands down a rope, and you say, oh, this is a spot that's weak in the rope, so you'll add some more duct tape around it, or make it sturdier. That's where the tRNA comes in and attaches on. It's got the amino acids. And so then step 2 we have here is called elongate. As the ribosome move along the strands of mRNA, each new amino acid carrying tRNA molecules binds to the next 3 bases. So it goes 3 bases, binds. Next 3 bases, another tRNA comes in, binds its amino acid onto the first one. Then next 3 bases, another tRNA comes in, binds its amino acid onto the next one. So we're making this chain of amino acids. When we get this long chain of amino acids, then we have our protein chain, which you can see right there. They kind of look like if you had a long chain of Skittles. And then imagine if they were all wound around one another. Or, you could kind of also look at this as, if any of you guys ever made popcorn chains at Christmas time, where you string the piece of popcorn onto the rope, and then you string the next one on, and then you put a cranberry on, and then you put another piece of popcorn, and then a Fruit Loop or something like that. So you're building this long chain of amino acids. That's this long chain of popcorn. It's got this 3D structure to it. And if you were to take it, you could wind it all up together into a popcorn ball, or you could unwind it and it would be a long strand, but it's this 3D structure. So the final step in this is that something has to tell it it's done. Where do we know that the protein chain is finished? And so that's the terminator region. Once the ribosome encounters the 3 base stop, so 3 letters tell it, all right, stop coding, stop putting amino acids on, release the chain of protein. And the protein assembly is complete, translation ends, and both the protein and the mRNA are released from the ribosome. So they're off to go do this process again and again multiple times. So the same mRNA molecule can be translated over and over again before it's degraded. So imagine this like you got your grandmother's cookbook and you look through it so many times and you're covered in sticky stuff from baking all these recipes, so the pages get all crusty and eventually they get all crinkly, and sometimes the pages fall out or they get covered in stuff and you rip them out. So this mRNA works for a really long time. Your grandma's cookbook's been around for a long time, but eventually it's degraded enough that maybe you can't read the recipes very well, or you lose pages, and that would be mutations. So it produces dozens, even hundreds of molecules of the protein until it's so messed up that it can't do it anymore. So how translation works, let me get the...
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome.
You know what, I'm going to pause that right there because I want you to make sure that if you don't have that in your notes that you put that in your notes. So the 3 base pairs, which are the codon, the triplet that I told you about, the tRNA comes along and it's got what's called an anticodon at the bottom of the tRNA. So it's like a set of teeth. You've got your codons, which are your 3 base pairs, and you've got your tRNA, which has its 3 base pairs, which are called the anticodon. So codon is attached to the mRNA, anticodon is attached to the tRNA. They fit together very specifically, very tightly, like teeth on a bicycle chain. They fit together like that, and then the amino acid is made and it joins to the next amino acid, to the next amino acid, to the next amino acid. So make sure that you know that, because that's something that's kind of important. You've got your codon on the mRNA, you've got your anticodon on the tRNA. They fit together like a very specific set of teeth and allow those amino acids to bind to one another.
Nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site, as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Are you guys cool with that, or do you want to watch it again? All right, I'll assume that means you're cool with it. And like I said, I'll post the links to these videos so that you can go through and watch them again if you would like to do that.